And in case of animal tissues, good morning kids. So today we are supposed to deal with study of permanent slides. Permanent slides and how to study them. Like in the previous experiment, I had shown you the microscope. The structure of the microscope we are very well aware of already. This is the eyepiece wherein we are supposed to keep our eye to observe the slide that is there on the stage. Here, there is no slide till now. Now, how to put the slide? We are simply going to remove the clips and we are going to put the slide in such a way that the circular part coincides with the circular part where the material lies, that circular part coincides with this circular part, and we are going to fix up the clips from both the sides so that the slide doesn't move. And we'll start observing from top and we'll adjust it with the coarse adjustment, the rough or the major adjustment. And then after that, we'll go for the fine adjustment. And with the help of this, we are going to study all the slides one by one. To begin with, the first one, plant tissues, 3 of them we are supposed to study. The first one is parenchyma. The second one, collenchyma. And the third one is sclerenchyma. And in sclerenchyma, basically, sclerids and fibers are there. As the steps indicate, for all the 3, we are going to have a look at the steps. If you are holding the practical book, well and good. If not, let's have a look at these steps. What all do we observe? So here we go. Plant tissues, and first step is for parenchyma. And if this comes in the examination, we are supposed to write down all the characters like this, that they are isodiametric, intercellular spaces are there, and there is a large central vacuole with a peripheral nucleus, and they are present in the soft parts of the plant, and their function basically is storage and photosynthesis. Collenchyma, on the other hand, somewhat oval to elongated, large central vacuole. As any plant cell, these features are common. Thickening at the corners of the cell, and that comprises of cellulose and pectin. This is the important feature of collenchyma. Of course, intercellular spaces will be absent because here the spaces are filled with the thickening, making sure that the thickening are there only at the corners. It is not covering the whole body of the cell membrane or the cell wall. Moving ahead with sclerenchyma, we have sclerids and fibers, and here of course we have mentioned for sclerids. Basically, mature cells are dead cells in case of sclerenchyma. They have thickened wall. That means all over the cell wall there is this thickening of lignin. So, of course, they are isodiametric. They may have these pits. These 3 we are going to study right now, one by one, of course. And let's have a look at how this parenchyma slide is going to be visible to us under the microscope. Under the microscope, diagrams we have seen, but how this slide appears For that, I have taken up certain pictures which make it very, very clear to all of us. And let's see. This is what parenchyma cells look like under the microscope. Now, if we observe carefully, the parenchyma cells have these thin walls. As the cursor moves all along the length, whole of the body of the cell, you can say that these are thin-walled cells. And this is how the parenchyma slide will appear to us. Almost similar to the diagram that we have seen. And now, let's see for a few more snapshots which are going to make us understand. After parenchyma, we are going to take up one more slide of collenchyma, and let's see how it looks like under the microscope. That means diagram, of course, we know, but how the slide basically appears to us in microscope, because right now we cannot have a live demonstration of the microscope. So here, let's see. This is how collenchyma appears to us. Clearly has been indicated collenchyma, and you can see the cell endings. Now my question coming to my mind is, will it be visible this way in the compound microscope? That means the microscope that I had just shown you. The answer is no. It really needs magnification, further magnification, and with that only you can see such thickenings very, very clearly. The slides in the school can show you some thickening, but this intense clarity will be there under even more magnification. So here you can see that the cells, and around the cells, the corners where three cells, for example, these three adjacent cells are there, and the ending of the cell is filled with the thickening. Of course, we know that that is, of course, cellulose and the pectin in case of collenchyma. Please, kids, try and observe. It may appear to you that whole of the cell is surrounded by the thickening, but it is not so. We are going to compare sclerenchyma and collenchyma also in one of the slides. But here, if you observe carefully, it is only the thickening where the adjustment cells are attaching. There, they have this thickening to fill that. So automatically, there is no intercellular space. Once again, the thickening is there between the cells, not on the walls of the cell, as it happens in case of sclerenchyma. And now let's see. Now we are going to understand for parenchyma and collenchyma. I am sharing one more slide with you. This particular slide clears the confusion between parenchyma and collenchyma. See, these are parenchyma cells, wherein the cell walls are thin. They are very, very thin. And here in the collenchyma, we have covered in case of a particular diagram in the book, wherein it says epidermis, hypodermis, and endodermis. So here in the hypodermis, we have this collenchyma, wherein there is this thickening is there in between two adjacent cells. This thickening, once again I repeat, is outside the cells, wherein the adjacent cells, they interact with each other. Now let's see for Sclerenchyma. And here I'll be comparing Sclerenchyma with that of other cells. So first of all, let's have a look at this Sclerenchyma. This is how the Sclerenchyma cells will appear. This red structure, you can say, this is the slide of it. And in originality, even more magnified, this is how the Sclerenchyma cells would be. All around the cell wall, all around the cell wall, there is this deposition of lignin. Therefore, we call them as lignified cells. Lignified cells. Now we are going to take up one more, which is going to make the confusion clear between Collenchyma and Sclerenchyma. Collenchyma and Sclerenchyma. Let's see. And this is how Collenchyma and Sclerenchyma is going to appear to us. It clears the confusion that here lies Collenchyma, wherein there is thickening. Here it is very clear where I have kept the cursor. Here it is very clear. Well, anyways, in all this particular area, in this particular area lies the Collenchyma, wherein the thickening is there at the end of the cells, and Sclerenchyma is like quite clear, ultra clear in the particular gap. This one holds the Sclerenchyma in the slide. Now, there is one more I have with me, which is going to make all the three cells clear. Let's have a look at that particular slide also, kids. And here we go. So these are basically the cells wherein parenchyma, both the sides you can see. Then we have this collenchyma, it's very clear here. And sclerenchyma. Sclerenchyma is like, you can say it's like odd one out. You can clearly demarcate their presence. So this is how the slides, highly magnified slides will appear. To some extent, we can magnify in school also using this compound microscope. But this is how the cells will appear to us in case of sclerenchyma, collenchyma, and parenchyma. Now you can put up in the comment box and tell me, have you seen all the slides very carefully? Is there any confusion in the slides of the plant cells? So that we can move ahead with that of the animal cells. All the slides were clear to all of you? Okay, good. Now, once again, good. I got your message, kids. I got it. Okay, good. Now, my request to you people is that please make sure that you are reading about them. In exam, how are they going to ask you? They are going to put up questions out of this. One particular characteristic feature they may ask you. They may ask you about the comparison between the two. As far as physics, chemistry, and bio are concerned, 2 mark each. 2 mark each. Take my word, it happens this way only. Year after year, half yearly and annual. 2 mark question from bio, 2 mark question from physics, and 2 mark question from chemistry practicals will be there in your paper. And unless you have seen the practical, you cannot write those answers. Practical is such a topic wherein as long as you don't have a demonstration, you have not been a part of it, you won't be able to write the answers properly because it's not theory. So please, my request to you is, we have the practical books with us. Read and write about this practical today itself. Make a PDF and upload it in time, in the due course of time. The assignments have already been uploaded. We'll now move ahead for animal tissue. Permanent slides, of course, animal tissue. And the way of fixing up the slide is as same as the one we have fixed up for the plant cells. For animal cells, of course, in our syllabus, we have all those that are mentioned in our chapter number 6, tissues. This practical is also related with chapter number 6 only. Okay. So what all animal tissues are we supposed to study? First is striated muscle fibers. Then second one is unstriated or smooth muscle fibers. And the next one is cardiac muscle fibers. And last but not the least is, of course, the nerve cell or the nervous tissue. And it's so very well clearly been seen in those slides. If you see the striated muscles, the voluntary muscles, the muscles that are present in our hands and legs, the appearance we have already covered in the theory. They have prominent light and dark striations and cylindrical in shape, multinucleated. Smooth muscles, spindle shape, tapering at both the ends, and uninucleate. Involuntary, present in the digestive tract. Cardiac muscle fibers, almost similar to this, but it will be branched. And of course, striations are very light, and this is going to be uninucleate. Nerve cell, long thread-like. Cyton, axon, and nerve endings. Here we have covered in case of nerve cell that one cell connects with the other one, nerve ending of one connects with the dendrites of the next, and the nerve impulse, the information, always will follow from head to tail. That is cyton to axon. Now let's start viewing, and here we have to view these slides very, very carefully. I'll be showing you those slides and you are going to compare their structure. All the three muscle fibers, initially the slides will appear as though there is no particular demarcation or difference between the three. But you have to be a very, very careful observer. Please observe the slides I'm sharing right now with you. The pictures of the slides, I must say, striations and many nucleus. I'll be keeping my cursor on those places. Please try to observe very carefully. And once we are talking about the smooth muscle, in smooth muscle, you have to see very carefully that the tapering ends are there and the cell carries only one nucleus. In case of cardiac, clearly demarcated, those branches are coming out, and that is uninucleate structure is there. Let's see first of all these three muscle fibers one by one. And here we go. And here I share the slide for the striated muscle fiber. Try to observe carefully. Try to observe very, very carefully. Striations. See this striation, this striation. And see, there are so many nucleus. 1, 2, 3. If you keep counting, there are so many. These dark small circles, they are the nucleus. And of course, here also the bands are very, very clear, light and dark. For example, dark and then light, then dark and then light. Same. From here to here, you can see this is a light band, then again the dark. Here also the dark and then the light. And here also goes the light. And even more clearer, the darker ones you can see. Here they are, for example, light and dark. Very, very clear. Very, very clear. This particular one area, if you try to observe very carefully, see light, then dark. Then here light, then again dark. Then light, it goes this whole length of the muscle fiber. Same happens here. You can see this graph like thing going on, and here, say for example, here it is dark and then the light portion. So these are striated muscle fibers, or the skeletal muscle fibers, or the muscle fibers that are voluntary, present in our hands and legs. Now I'll be sharing with you the cardiac muscle fiber. The cardiac muscle fiber I have put first before smooth, because you need to compare branching. Here you can very well see the branching. Observe carefully the branching that it has. See how nicely it is branching. The single fiber is coming, and here it is branching here, and then it goes here. And every muscle fiber, you can say only one nucleus. For example, from here to here, this one is very, very clear. From one band to the next, only one nucleus. Light and dark striations, but not that dark. It has been stained darkly. And here we have this dark and then light band. And moreover, it is branching. You can see here also it is branching here, and here it goes again. And this one also is branching. One branch this side, another branch that side. This one is also branching. Here it goes, and here also it goes. This one also is branching on both the sides. So that is basically the branching of the cardiac muscle fiber. And now, let's see. I'll be sharing with you for smooth muscle fibers. Let's see if we can have a look at that also very clearly. This is the slide for smooth muscle fiber. Now, how to identify whether it all appears the same only, all the three? But here there is no striation, and please try and observe the structure. See, this white portion is so clear from here to here, and then it goes back with a single nucleus. Again, this portion is actually very, very clear. Well, all of them, they are the same, but this has been like separated or segregated so nicely. See this one, tapering end on both the sides and one nucleus in the middle. And this one, this you can say a magnified way, you can say the bigger one they had shown up here. When you try to focus on these, for example, this one is so clear, tapering on both the ends, and here it has the nucleus in the center. Same happens here. But here on this side, the tapering is not that clear. This, or you can catch hold of this one also where I'm keeping the cursor, tapering on both the sides with one nucleus in the center. And again, tapering on both the sides, one nucleus in the center. Tapering on both the sides and one nucleus in the center. And this is how the smooth muscle fibers will appear to us under the microscope. Highly magnified, of course. And now I'll be sharing with you nerve cells, some of the beautiful slides I have got. And this is how actually the nerve cell should appear to us. Nerve cell highly magnified. This magnification is not possible with the compound microscope, but I wanted you to have a look at this picture. And let's have a look at this one. This is how the nerve cell actually, the slide diagrams, of course, we have seen, but this is how a single nerve cell appears to us. A single nerve cell. This is the cyton with lots many dendrites around, one after the next, and there it follows the axon or the tail of it. And this cyton has one nucleus in the center. That means the cyton lies from here to here, where I'm keeping the cursor. And there continues the axon or the tail of it. Let's see how these are actually connected. One more, this is the type of the particular slide we'll be seeing in the school. But one more thing, which is like very, very beautifully depicted, the connections of the nerve cells. Let's have a look at that. It's such a beautiful, wherein you will be able to understand how the nerve ending of one joins with the dendrites of the next cell. This one. It's a beautiful depiction. Try and observe very carefully. See how this one connects. It clearly says the processes, axons and dendrites. Say for example, here lies the axon, the cell body, and its tail is continuing, and it connects to the next cell at this juncture. Same, this appearing like star, this is another cyton, and its processes, that is the axon, follows and it connects. It's not that one cell is going to connect to one more cell only. One cell connects with so many, forming a network. So see how this one connects, one on this side, the other one on this side. This particular nerve cell has its dendrites coming over here and here, and it connects at this juncture. These are like different nerve cells, this one, this two, this three, and here you see the cell body very clearly. The cell body and its processes. This cell body and its processes, these are like dendrites coming out. All dendrites coming out. And these dendrites are joining with the axons of the other cells. Same happens with this one. This is also sending to some other cell, and here its tail, you can say, goes and connects with the other cell. So if you observe very carefully, there are all in all like very, very clear. If you see the connections are very clear in this case, 1, 2, 3, 4 and 5. The way they are connected, the tail of one, or the nerve ending is a proper term, of one connects with the head of the other. For example, these arrow marks, see, look at this one. Okay, one process coming from this particular one and connects with, here this particular cell has its dendrites going up. The dendrites from here, it joins with the axon of this cell. The processes goes on this side and connects with the other cell. So it's like a network, a whole network, wherein the dendrites are connecting with the nerve ending of the next nerve cell. And the nerve impulses go from one cell to the next. So that's all from my side for this particular practical, that is study of the permanent slides of plant tissue as well as animal tissue. And if you feel, I can share these slide pictures in your group also, so that you can go through them properly. Just you can comment in the comment box and tell me if you need these pictures in your group. I'll post so that you can have even a clearer idea about the slides, how they would appear to be. All right, I'll do that. I'll do that immediately after this class. I'll do that. I'll post all these pictures in your group. Moreover, try to explore more and more processes on it, clicking on for the slides for these particular tissues. Now, once the situations are normal and you are coming to the school, hopefully it happens this year itself, then in that case, I will be in lab showing you all the slides once again. There's no harm in repeating for the kids. That's all from my side, kids, for this particular one practical.
Ma'am, I have a doubt.
You had learned and try to read and understand more, and we'll discuss about the question answers related with these practicals in the next revision classes. Happy learning, kids. Stay safe.
Ma'am, I have a doubt.